Today, we're going to be talking about Darzalex, an antibody drug by Johnson & Johnson used to treat multiple myeloma.
Multiple myeloma is an incurable form of blood cancer that affects plasma cells located in the bone marrow. Healthy plasma cells help the body fight infections by making antibodies that recognize and attack germs. Cancerous plasma cells reproduce and replace the normal white blood cells with tumors in the bone marrow.
It affects over 34,000 people per year, and even with early intervention, only has 56% survival rate. It is a 1.5 times more prevalent in men, and treatment can cost tens of thousands of dollars.
Darzalex is a monoclonal antibody drug which targets melanoma cells while sparing healthy cells.
Darzalex works through protein inhibition of CD38 protein. This is a transmembrane protein found on all plasma cells, which helps regulate homeostasis and metabolic functions. It is overexpressed on myeloma cells, meaning there are many proteins for the Darzalex to bond to. The Darzalex antibodies are designed to recognize the chemical environment and interact with the CD38 protein.
A critical interaction between drug target and antibody residue is a cation pi bond between the heavy chain phenylalanine 32 antibody residue and the lysine 232 drug target residue. A cation pi bond is a strong covalent bond interaction, which could be the main reason that the antibody stays bound to the drug target because it takes a lot of energy to break.
There are numerous hydrogen bonds between the antibody residue and drug target residue. The hydrogen bond includes a hydrogen donor from one residue and hydrogen acceptor from another residue. A hydrogen donor consists of a hydrogen covalently bonded to an electronegative atom. A hydrogen acceptor consists of an electronegative atom with some degree of negative charge. The two functional groups with each residue interact non-covalently to further bind the drug and protein. The orientation of the various interactions between the two allows specificity and binding, and positions the two molecules in such a way that it can inhibit tumor cell growth, causing myeloma cell death.
Darzalex can cause cell death in 4 different ways, several of which utilize the body's natural immune system. The methods are direct cell killing, antibody-dependent cell-mediated cytotoxicity, phagocytosis, and complement-dependent cytotoxicity.
Direct cell killing occurs when Darzalex antibodies bond to many CD38 proteins and cause clusters on the cell surface. This disrupts metabolic activity that normally occurs via CD38 proteins. It specifically prevents the transfer of mitochondrial from bone marrow-derived stem cells to the cancerous cluster of cells. Consequently, the prevention inhibits tumor proliferation. Through this mechanism, Darzalex can directly cause apoptosis or cell death of the cancerous plasma cells.
The second method, antibody-dependent cell-mediated cytotoxicity, occurs when Darzalex is bound to CD38 on the surface of melanoma cells. The stem-like portion of the Darzalex attracts and binds the immune cells. It bridges the target cell and immune cell, facilitating an immune response. An important cell it recruits is a natural killer cell. The natural killer cell is part of the immune system's innate defenses. Natural killer cells have the ability to quickly determine between healthy and abnormal cells and works in conjunction with other non-specific cells and molecules to kill unhealthy cells.
The Darzalex antibody can also bond to a type of immune cell called phagocyte. There are many different types with different functions. Macrophages and dendritic cells both function as antigen-presenting cells and can initiate a T cell response against cancer cells. In this case, the M1 macrophage would play a role in eliminating the tumor cells. When dendritic cells recognize danger, they gather and internally process nearby dying cells and cell debris. They then travel to the lymph nodes and activate a specific T cell that recognizes the particular antigen. This activation stimulates the multiplication of T cells to attack the threatening cells. In combination, phagocytes attack in a process called phagocytosis, in which they engulf the cancerous plasma cell, causing cell death.
The stem of the Darzalex attracts and binds to molecules of the immune system called complement that freely flow in the blood or tissues. These molecules work in a system to attack viruses, bacteria, or other threats and help to clear damaged or unhealthy cells. The system contains proteins that can kill certain threats, in this case cancer cells, directly or can flag them for removal by phagocytes. The cell that Darzalex is bound to is then destroyed.
Darzalex has shown to be effective as a single treatment as well as when combined with other forms of treatment. The treatment has accumulated over $2 billion in total sales and has been used to treat more than 227,000 patients worldwide and 68,000 patients just in the US since its approval in 2015.